The other thing that I wanted to bring up in this is the use of some very, very high-tech gene sequencing equipment. So this is the kind of equipment that when you read about the sequencing of the human genome or the cow genome, in DPI Victoria, we did some work at sequencing the endophyte genome, a much smaller genome, and what we found was that some of the new strains of endophyte that were being developed or soon to be released and are coming out now actually lacked the genes. This little here, so this says that's a gene sequence there. This gene here is involved in lolitrum biosynthesis, so the main compound that causes ryegrass staggers, and this yellow strain and this pinky purple strain lack that gene. They've lost it somewhere in their evolution. So we can be absolutely sure when those strains are released, no matter what environment, no matter what plant genetic background, they will never produce lolitrum. They lack the machinery to do it. If you compare that to those who have been around for the very first releases of novel endophytes with endosafe technology here some probably 15, 20 years ago now, we found that environmental variation did change the way that was expressed. So a number of breeding companies are now using this kind of technology to make sure that what they're developing is more robust. The other thing that we were able to do with this, just quickly to demonstrate this, was to go within a company's breeding program, and we demonstrated to them that they thought they had an animal-safe endophyte, which I'm calling strain X. When we actually looked, we found that they had two endophyte strains growing in the plant. So without this technology to know this, they wouldn't have been able to see this. And importantly, these strains were different in the way they performed. So I'll just scroll through this. Oh, it hasn't picked up the labels on the bottom, so I'll just quickly go through this. This is the presence of lolitrum here in the wild type, and you can see that the strains here produce less of it. But again, in another group, they've got this line C, which is clearly the wild type endophyte. So we were able to very clearly demonstrate that you could use this DNA fingerprinting technique as a very solid QA in a breeding program. We're fortunate that the endophyte, being a fungus, and particularly this fungus, has a very simple genetic background compared to the plants we're dealing with. So we've also then started to think about DNA fingerprinting technology in pastures. And James mentioned that sometimes this work is very long term. When we started this work, a very senior serial geneticist, a wheat guy, he said to me and my colleagues, "You will never get markers to work in forage species. It's too hard. They cross like animals. You've got multiple parents. You've got this issue of polyploidy I already talked about. You know bugger all about the traits you're really breeding for. Compare that in wheat breeding. We know which glutenins, which proteins, how the starch interacts. You guys have got no clue, basically, so you'll never make it work." We were fortunate that we saw it as a challenge, and that the government and Dairy Australia and other groups said, well, if we don't get it to work, one reason to be sure we won't get it to work is if we never try. So we had a decent go. And so what we did was pinch some ideas, not from plant breeding, but from animal breeding, to think about how we would use DNA fingerprinting to allow us to take advantage of those things and say, not every pasture plant that goes in a new variety needs to be the same. If there's some diversity out there, it allows them to interact in ways we don't understand that can result in improved persistence. They can result in those cultivars doing better in environments we don't understand. So rather than fall into the situation where barley breeders are today, where they have to sacrifice yield to maintain malting characteristics, and it's one of the reasons why if you ever try and buy a malting barley, there are very few of them that will yield the same as a feed barley, because the whole breeding program is driven by that complex thing which is malting quality. Now, we did some of the work to develop the first genetic maps of perennial ryegrass. So this looks like, you know, some kind of fish skeleton with all the bones coming off. But what it is, importantly, is a genetic map of perennial ryegrass chromosomes, and all these coloured genes here are genes involved in lignin biosynthesis and forage digestibility. So we're able to develop really strong targets for our DNA fingerprints to find the plants which have higher quality, either because their cell walls are more digestible or they have more carbohydrate content. We then use this work to understand the diversity for one of these genes, which I won't say its full name, but it is a gene involved in lignin biosynthesis, and it's a particularly bad gene because what it does is it binds lignin to the fibre of a plant and makes that fibre unavailable to an animal. So we're looking at finding good variants of that gene in terms of animal performance. And as I said, if you don't see these technologies in isolation, so if you say, oh, we're not going to have a research program with GM because we're not certain about the political climate 20 years out from today, you wouldn't do work like this. And so this is a standard ryegrass stem, so you can imagine it's round. The coloured staining here represents lignified tissue. This is tissue which is not available to digestion by an animal. You remember from the previous slide, the gene was called CCR1. These are two experimental perennial ryegrass plants where the gene for CCR1 has been switched off using genetic modification, and you can see that the amount of lignification has been greatly reduced down to very low levels. And the one place where you see that it still remains are these little circle bits here, and that's actually the vascular tissue. That's where the ryegrass pumps the water up and down, so we actually want that to remain intact. It is important to remember in all of this that the development of these plants is the first step. Then they get delivered through breeding programs. So this is an example of a breeding nursery of mine from some years ago now. At the time, these were all commercially available cultivars of perennial ryegrass sown as individual plants at Hamilton. This is regrowth after autumn rain. This is a slide that I've borrowed from AgriSeeds in New Zealand and represents their ryegrass breeding program, but they're pretty typical. So you can still see that we're looking at somewhere in a 10-year cycle. So what we're trying to do is to use these DNA fingerprinting tools to drop out some of these years or to allow breeders to focus on traits which have a higher value. So even if it still takes 12 years, the rate of gain coming out at the other end is higher. The final example I wanted to touch on before we can have a bit of a discussion was some work we're doing with white clover and using genetic modification to develop better white clover. We all know that the amount of white clover in dairy pastures has declined dramatically over recent years. We also know it's much harder, no matter how much we want to maintain white clover in the system. One of the reasons for that is that white clover has some genetic limitations. It is susceptible to a range of diseases. There are issues with the quality of white clover and its biomass compared to grasses. The seed production in white clover can be very low, but there are some advantages in terms of looking at how we can make white clover more tolerant of acid and using it to better acquire phosphorus that's bound in soils. But the work we started with, and again this is a long time ago, was trying to look at this alfalfa mosaic virus. And really it was an intractable problem for us. It has a very high incidence in pastures. If you go and survey, almost all pastures have it. This is the symptoms here at an early stage. You can see here this is a sick leaf versus a healthy leaf. There's a whole range of things it causes, but basically it makes the white clover yield less, survive less, and importantly, nobody has ever found any genetic variation for resistance to this virus in white clover. You never say never in biology, but lots of people have looked and nobody's ever found it. So we used a GM approach which is similar to vaccination in that when this virus comes to infect the white clover, it sheds what's called its coat protein and then infects. If the virus detects that the coat protein is already in the plant, then it says, okay, this plant's already infected, I'll just keep alive to infect the next one. So we use that technology in essence to disable the virus, so I don't need to go into this. And over a number of years, what we were able to show, so you can see that these are field trials of white clover in Hamilton in 1998, so it's been around a long time. And over time, this is the level of virus in the control plants, and this is the level of virus in the genetically modified plants. Happy to say that that's been confirmed in a number of backgrounds and a number of crosses over the years. I won't go into that. Again, I just want to make the point that anyone who gets up and says any of these technologies, snap bang, it's going to work overnight, they're not overnight fixes. So there is the need for a very complicated crossing program, but the drive is to do something here that you couldn't do otherwise, to get resistance to this disease, which was a problem. I can tell you as a plant breeder, and I did these crosses, I would never have taken this GM path if I could find resistance in another way, because I would knock out all those lab steps and get into a much less complicated crossing program. But the one thing I want to say is that in this genomics biotech area, persistence does pay. In that white clover example I showed there, looking at 10 years of research and development that has a whole range of sciences and a whole range of issues that we're not so used to as plant breeders in terms of dealing with the compliance management of a government regulator. In Australia, all work with genetically modified organisms is governed by the Office of the Gene Technology Regulator in the Department of Health and Ageing in the federal government, so it changes the way you do science. And a commitment from partners to go the journey to say, well, what could this science do? Not necessarily we absolutely know this science will get on farm, but understanding what it can do. And this material now has been undergoing commercial trialling pre-release by the Victorian Government's commercial partners in the work. So what I just want to conclude with is that what I'm passionate about is the conversion of knowledge to products for farmers and getting the right alliances along that supply chain to ensure we get there in the end. And I'd just like to acknowledge a whole range of people who've done this work with me over the years. Herman Spangenberg and John Forster have been my colleagues in this work for more than 10 years now. Arturo was an excellent PhD student of mine who did a lot of the white clover work, and a whole range of colleagues within DPI and collaborators in industry. And as I said, the work that was presented here was done while I was working with DPI. So thanks, Rick.